Today, I'm very happy to discuss with you about how various factors impact the polygenic prediction across populations. So my presentation today will cover those different sections, and I'll pause after each section for questions. So to start with, I'll give a brief overview of PRS. PRS use the data obtained from GWAS. So in the past years, GWAS have witnessed a massive success that have identified thousands of variants associated with various diseases. And there are many applications to translate the findings from GWAS. One of such application is PRS. PRS, they are actually predictors of the genetic susceptibilities of individuals to disease. Its calculation is quite simple. It's the sum of the risk alleles weighted by the effect sizes. So by its format, we can say it has two key components. The first one is the number of risk alleles, usually coded as 0, 1, or 2, as illustrated by a single variant here. So the second component is the estimated effect sizes from GWAS. So this tutorial example shows assuming an individual has 100 risk variants and each with equal contribution to the disease risk. So by summing those risk alleles, the PRS for this individual is 21. So this illustrates how we can calculate PRS for a single individual when extend to a general population. So assuming we are interested in a case control disease endpoint, we can see their PRS pattern greatly varied between diseases and controls. So for those cases, we can see at any particular locus, they may not carry that risk variant. And for the controls, we can see they can carry some risk alleles at some locus, some loci. In general, the distribution of PRS, they follow a normal distribution in both controls and cases. But for the cases, they have a shift towards the higher number of high-risk alleles. So knowing the distribution in the population, we are wondering how we can run PRS analysis. So the steps is quite simple. As we just mentioned, it starts from GWAS. So we perform GWAS in the training population to get the estimated effect sizes. Due to the technical issues such as limited sample size of the GWAS, the effect sizes, they are always estimated with errors and they may suffer from venous curves and other issues. So we usually run different PRS methods to reshrink or rescale the effect sizes. So those methods are defining these two main components, like which variants are included and what weights are associated with these variants. After we running different PRS methods, we will develop the PRS in the independent test sets. To evaluate the performance in the test sets, there are quite a few different metrics. For the quantitative traits, it is quite intuitive. We usually use the regression coefficient, which is the R-square from the linear regression. But for the binary traits, there is no such R-square. We can use some shorter R-square, but more often we are using the R-square on the liability scale to account for the disease prevalence. And also we may report the AUC or C-index with values of 0.5 or 1 means there is no or perfect power to distinguish the cases and controls. So knowing how and what is PRS, then we may wonder where PRS could be applied in the population cohorts. So a nice review from Ray et al. They summarized a few scenarios where PRS can be applied. The first scenario is related to disease screening, like assuming disease have a lifetime risk of 1%. then for those individuals in the top PRS stratum, they are having a higher probability of getting that disease in their lifetime. And they are extremely encouraged to enter the established disease screening. So this scenario is mostly useful for disease like cardiovascular disease, breast cancer. And it's kind of inspired by a study performed by Dr. Kohara et al. In 2018. So in this study, they constructed PRS for the coronary artery disease, and they found they can identify 8% of the population at a threefold increased risk for coronary artery disease. This prevalence is actually even higher than the monogenic mutation, conferring the comparable risk. So another study showing the coronary artery disease-based PRS, they can actually perform much better than the individual conventional risk factors. And further integrate PRS with the conventional risk factors, they can achieve better predictive performance. So another use case for such risk stratification is breast cancer. So the Bolivia model for breast cancer integrates different levels of risk factors, such as family history, age, where the genetic risk can integrate both real variants and common variants from the PRS. So this combined model can stratify the individuals in the population to different levels of risk. So for those people with high risk, they are encouraged to have a breast screening at an earlier age and also maybe more frequently. And a few applications of this model have even shown PRS have higher predictive power than all the combined conventional risk factors. This highlights again the utility of PRS for risk stratification. So translating PRS from discovery to the clinic may involve different processes. To date, there are a few clinical trials on PRS. Here, I want to mention the one conducted by Genomic Medicine at Veterans Affairs as an example. So this trial, they are focused on traits like breast cancer, prostate cancer, and type 2 diabetes. They summarize the whole trial as 3 different phases. In the first phase, it's pretty much the same as what we are talking about, the analysis steps for the PRS. And in the second step, the laboratory, they will develop a clinically valid assay. And in the third step, the treating physician, they will integrate all the patient context to interpret the significance of the PRS for the patient's health care management. So these are a few cases for how PRS can be applied for disease screening. Another scenario is that when the cohorts, the people are presenting with some clinical symptoms, but there is no clear diagnosis. In this scenario, PRS can be used to guide monitoring of emergent symptoms, such as like maybe around 15% of adults, they are presenting with type 2 diabetes, but are misdiagnosed with the more common type 2, they are presenting with type 1 diabetes, but they are misdiagnosed as the more common type 2 diabetes. So within those with type 1 diabetes, if they are having a high PRS for type 1 diabetes, they could be used to trigger more frequent monitoring of insulin levels, because type 1 diabetes PRS, they will find to predict the progression to the critical phenotypes of the insulin deficiency. So another scenario for the use case is all the individuals, they are diagnosed with this disease, but PRS, the genetic information contained can help to enable a more effective choice of treatment and to reduce the adverse effects. So far, we have introduced analysis steps of PRS and how they can be applied in population cohorts. Here, I want to highlight that there is a growing recognition of the need for equitable use of PRS in ancestral diverse populations. This is especially important for countries like US, where there is a significant diversity of ancestral backgrounds. So it is kind of a hot research topic, polygenic prediction across populations. Before starting this section, I would like to take a pause to allow for any questions that may arise.
There are no questions in the Q&A right now, but one of my questions is, and you spoke a little bit about this, but if someone is just reading a polygenic risk score paper, what are the most important quality metrics you think for people to look at when assessing if the PRS is likely to kind of capture a significant proportion of disease risk?
I think that's a very good question. So the metrics, they may vary depending on the type of traits. And also the research question, like I just mentioned, so the R-square, they are more comparable like, to the SNP-based heritability, which actually is the upper bound of the PRS accuracy. So if we want to evaluate how the PRS compare to the trait heritability or SNP-based heritability, we may use R-square or shorter R-square or R-square on the liability scale to report the performance. But on the other hand, like in a more clinical setting, like people are more interested in metrics such as AUC to say is discriminatory power of the model. And more often, people want to report like odds ratio. Yeah.
That's so helpful. Thank you so much.
Okay, so I will continue on polygenic prediction across populations. It is well acknowledged that the PRS generalizability is limited across populations. As shown here, when using PRS derived from European GWAS, its accuracy is much attenuated in the non-European populations. So this limited generalizability is largely due to the fact that current genomic studies, they are predominant by European populations. So this limited transferability or generalizability are due to many factors, such as the demographic history and the between ancestry differences in the manual allele frequency and LD patterns. So to improve the PRS generalizability, we may start by looking at what and how different factors affecting their theoretical expectations. So when we are predicting within the same population, that's to say the discovery and the target population is the same, then the theoretical accuracy is a function of the SNP-based heritability and the sample size of the discovery population, also the number of the genome independent chromosome segments. But when we are doing like polygenic prediction, there will be more factors involved, including factors such as the cross-population genetic correlation, and we also need to account for the heritability in the target population. So based on those theoretical expectations, we may see different strategies to improve the PRS accuracy and hence the generalizability. So first of all, we can increase the sample size as well as the diversity of the discovery dataset. And secondly, we may use some more advanced statistical methodology to improve the accuracy. Those methods can be used to like generate optimal ways associated with the risk alleles and also select more variants that are truly associated with the phenotype of interest. So for the first point, there are many ongoing efforts to improve the diversity of the current genomic studies, such as ORFAS, GIANT, NOMAD, etc. But here I would like to use Global Biobank Meta-Analysis Initiative, or GBMI, as an example to illustrate how we can collate across existing boundaries. So this work led by Dr. Weizhou, we collected more than 2.2 million samples with both genotypes and phenotypes from 24 biobanks, and the number is currently increasing. So they are from different origins and ancestries. For each participant in GBMI, they are assigned to one of these 6 different populations, ranging from like 1.6 thousand Middle Eastern to up to 1.4 million Europeans. So to illustrate the PI's work in GBMI, I'd like to take asthma as an example. So asthma is one of the largest endpoints in the 14 endpoints previously studied in GBMI, and it also has wide prevalence across different biobanks. So we construct PIs based on GBMI GWAS versus the previously largest asthma GWAS from TAGC, and we test the accuracy and report the R-square on the liability scale in different biobanks. So the x-axis shows the target population in each of the biobank. We see like the GBMI GWAS-based PIs, they actually improved a lot of the accuracy across different ancestry populations. So these results highlight the power of increasing the genomic diversity of the discovery GWAS, as well as like the benefits from the larger sample sizes. So another strategy I previously mentioned to improve the PI's generalizability is to use more advanced methods. So in our review, we summarized a few methods that like leveraging the shared genetic components between ancestries, and also these methods can incorporate additional data from like functional annotation or fan mapping. And also we can use multi-trait methods that use the pleiotropy across different traits. So to illustrate how those methods can improve polygenic prediction across populations, I like to use PICSX as an example. So PICSX, they use inputs of GWAS from different populations, and they can jointly modeling the between ancestry, LD, and minor allele frequency differences, which is a basic method. So in their paper, they compared PICSX with a bunch of other methods and report the R-square on the liability for the schizophrenia across a few East Asian cohorts. So in the x-axis, they use different methods, like either based on the East Asian discovery GWAS or European discovery GWAS, or use those both GWAS from these 2 ancestries. So among all these comparisons, they found PICSX actually show the best performance. So now we have showed how different strategies, either improving the diversity or use advanced methods, can improve the PI's generalizability. But few studies have explored how this could be affected by various factors, such as ancestry, genetic architecture, and PI's methodologies, especially in the context of leveraging GWAS from multiple populations. So any questions before I start on this section?
Not at this time, thank you.
Cool. So to explore how those factors affect polygenic prediction across populations, our study design follows like the general PI's analysis step we just mentioned. So first of all, we run GWAS in the discovery population. Here we focus on both single ancestry GWAS, which is performed on one ancestry, and also GWAS combining more than 2 ancestries. So for the single ancestry GWAS, we are using European from UK Biobank, and the minority population we are focused on here is East Asian from Biobank Japan. So for each individual population, we randomly split the whole population into many equally sized bins, with each bin have 5,000 individuals, and we run jewels in each of these bins and then meta-analyze different number of bins. So the sample size of the discovery jewels will be different. So to mimic the multi-ancestry meta-analyzed jewels, we meta-analyze European jewels and the biobank Japan jewels and vary the contribution by changing the number of bins from each population. So the ancestry composition of these meta jewels will also be different. The second step for PIS analysis is performing different PIS methods. So in this study, we are using, like, except for the previous mentioned PIS-CSX, we're also using P+T and PIS-CS, which we will mention a bit more in our later slides. And the final step is that we construct PIS in the test set. In this study is the East Asian population from UK Biobank. So the classic P+T or pruning and thresholding methods, it involves two processes. They can, the first process is p-value thresholding. Like if you set p-value thresholds, so any SNPs with a GWAS p-value smaller than that threshold, they will be included in the process. And the second process is the LD pruning or LD clumping based on predefined LDR² thresholds. So any variants that are in LD with the most significant variant in this LD block, they will be removed. So only those SNPs highlighted here will be retained. This process will be iteratively run by the next most significant variants until there will be no variant selected. So it's actually a kind of method to select a subset of quasi-independent genome-wide SNPs. Where PIS-CS is a kind of more advanced Bayesian methods, it can joint model all the genetic markers without specify any arbitrary thresholds for p-value or LDR². Here I won't go any details for the methods, but I want to highlight one key hyperparameter here, which is the polygenicity. So it is the proportion of SNPs with non-zero effects and is quite related to which variants will be included in the PIS model. So if the trait is less polygenic, then we expect there will be fewer variants be included. But if the trait is more polygenic, then we expect there will be more SNPs included in the PIS. So the traits we are focused on, ranging from anthropometric traits such as height and BMI and a few blood panel traits. So the sample size varied between biobanks and also across different traits. To get some information on the genetic architecture, we estimated 3 key parameters here. One is the polygenicity we just mentioned, and the second is the SNP-based heritability, where in the theoretical accuracy, we show it actually is the upper bound of the PIS accuracy, and the self-parameter is S. S is a measurement of the relationship between the allele frequency and effect sizes. So if S is negative, it can indicate the trait is under negative selection. So across those 3 estimates, we can say the genetic architecture of those study phenotypes greatly varied. For the traits like MCHC, MCH, and MCV, they are showing quite low polygenicity as compared to traits like height and BMI. Also, comparing BBJ in red and UK Biobank in blue, we see the estimates of both polygenicity and heritability is lower, which are likely due to the sample size in BBJ is also much smaller. So after knowing the genetic architecture, we then started to construct PIS. We first construct PIS using single ancestry jewels. So we used ancestry matched GWAS, which is BBG, as we are testing in the UK Biobank East Station. And another set of GWAS is European from UK Biobank. So we are using two different methods with P plus T is the light color and PRCS is the dark color. So the information I want to highlight here is that for those traits using ancestry matched GWAS, even the sample size is much smaller compared to the large-scale European. Their accuracy is comparable or even better. So as pointed here for height, both P plus T and PRCS in BBG, they are performed comparable to large-scale European GWAS. And we found the same for MCV and MCH, but only for P plus T. So I need to highlight here, like as I just showed, these two traits, they are presenting quite a sparse genetic architecture, like the polygenicity is small. So we then constructed a PRS based on the multi-ancestry meta-analyzed GWAS. So we call this PRS multi. So for such meta-analyzed multi-ancestry GWAS, we may use a different LD reference panel to approximate the LD information for these SNPs. In this study, we tested 3 sets of LD reference panel. The first one is a combined LD reference panel, which have the sample size, they are proportional to the ancestries represented in the multi-ancestry GWAS. And other 2 single ancestry GWAS, including East Asian and European. So in this figure, the y-axis is the accuracy in the East Asian, and the x-axis illustrates how the ancestry composition of the multi-ancestry GWAS. So in this PRS multi, the European GWAS is 322,000, and the sample size from the Biobank Japan included in the multi-ancestry GWAS is shown in the x-axis. So we can see in this scenario, the European population is dominant in the multi-ancestry GWAS. So we see the accuracy using combined or European as the LD reference panel is very similar, showing the impact of LD reference panel in this scenario is very slight. So after constructing both PRS multi and PRS single, we want to compare these 2 different PRS. So the y-axis here shows the accuracy differences between them. And any point above this line showing the accuracy using PRS multi is better. And similar for this below the line showing the PRS using European GWAS is better. And in these cases, we are reporting like the European GWAS with this 322,000 GWAS sample size. So first of all, we compare like around 3,000 comparisons. We showed for both methods, the majority of the comparisons where PRS multi actually outperformed using the large scale European GWAS only. And specifically from the figure, we can see this accurate improvement of PRS multi, they actually increased when the x-axis, which are the sample size from East Asian, increased. And we showed here the largest improvements using those 2 methods for the 3 traits. So often, like when we have a GWAS from multiple ancestries, another common way is that we construct a single ancestry GWAS based on each of the population. And we then using a linear combination to combine these PRS through an optimal weighting strategy. So we refer this strategy as PRS weighted. So in this analysis, we compare like the previously meta-analyzed multi-ancestry strategy, which is PRS multi, with this PRS weighted for these 3 different methods. And this y-axis is still the accuracy in the East Asian. And we showed the same figure also for other traits. to better illustrate how they show across different trees. And overall, for those 3 methods, when using 2 different strategies, we found out of all the comparisons for P plus T and the PRCS, using meta-analyzed strategy actually performed better than the weighted linear compilation strategy. This is not true for PRCSX. And another information I want to mention is that when we are comparing those 3 methods, like PRCSX performed the best than the other 2 methods. But there are one exception, which is BMI highlighted here. For BMI, the PRCS actually performed better than PRCSX when using the PRC multi strategy. And also we found that for the PRCSX, for the 2 traits which are less polygenic, as I just mentioned, for the MCH and MCV, using the weighted strategy actually provided better than the meta-analyzed strategy. So until now, like our discovery was focused on relatively homogeneous populations. So we want to see whether these findings can be generalized into mixed populations. So we then test the accuracy based on PRs derived from local ancestry informed GWAS. So here we are comparing a set of different discovery GWAS where the African tractor are the GWAS performed on the African tracts in the mixed African European individuals using the tractor method. And the other 3 strategies, those we just mentioned. One thing I want to highlight here is that we note the sample size of the African tractor GWAS is much smaller. It only have around 4.5 thousand individuals compared to other 3 sets of discovery GWAS. And we are focused on those 3 methods and report the accuracy in the African. So here is the figure like extended to other traits, and we found African tractor, like even with much smaller sample sizes, they provide actually comparable or even better accuracy when we are comparing to the European GWAS, which is now with a sample size 320,000. And these are 2 like for traits categorized by like WBC and neutrophil. They are with some African enriched variants where their allele frequency in African, and also the effect sizes are large. And also for traits like MCV, MCH, and MCHC, they are quite less polygenic with a sparser genetic architecture. So for those 3 traits which are less polygenic, we see consistent with our previous findings, P plus T actually performed better than the more advanced PRCS method. And also for those traits, for those 5 traits, we found the best approach for the local ancestry informed PRs is through weighted linear combination instead of meta-analyzed multi-ancestry strategy. So this is also consistent with our previous findings. So a brief summary for this section. In terms of ancestry composition, of course, we want to use ancestry matched GWAS. This highlighting like the necessity of increasing the diversity of current genomic studies. And when increase the the diversity of the discovery GWAS, we found the accuracy also increased for the PRs multi as shown in our previous slides. But we may not expect the accuracy will improve when we are meta-analyzing very small GWAS from the minority population, such as in the magnitude of 5,000 or 10,000. And in terms of the PR's methodology and genetic architecture, so we are for the For the cases where we are using resources from multiple populations, we found, especially for methods such as P+T, directly meta-analyzing those datasets, they will provide us a better performance than using the linear combination through a weighting strategy. And among those different methods, we compared basic methods. They are adaptive to a trait-specific genetic architecture, overall provide a better accuracy than the classic P+T methods. But there are few exceptions with these traits which are less polygenic, or they are showing some large effect ancestry enriched variants. So for those traits, we found using a weighted linear combination, they are better than a meta-analyzer strategy. And also P+T can perform better compared to basic methods. And also for those traits, we can use local ancestry informal GWAS, even though their sample size is much, much smaller, they can improve the PRS accuracy in the understudied populations. So before I go to the final section, any questions for the audience?
I guess when applying, I know you showed across a bunch of different traits, were there any patterns to kind of which categories of disease or which categories of traits were kind of best, best ported across ancestries, and then maybe other kinds of traits where you saw the accuracy was not as high when you were looking at the smaller populations?
I think that's a very good point. So in generally, like there are increasing evidence showing like the causal variants, they are shared across different ancestries and populations. So in these scenarios, those traits, their generalizability are mostly affected by LZ and the allele frequency differences between populations. So they are kind of in this similar level in terms of accuracy or generalizability. But for a few traits I highlight here, like the white blood cell counts or neutrophil counts, they are presenting. And also for GALs, in one of the endpoints we studied in GBMI, they actually, in some of the long European ancestries, they are presenting like higher allele frequency in that understudied populations. For those traits, even the accuracy may be higher. But in these scenarios, if there are some population-specific variants, the generalizability might be lower if they are used as the discovery GWAS.
Makes sense. So it's more kind of ancestry dependent and not kind of patterns based on traits or kind of organ system, things like that.
Yeah, that's true. Like everything is trait-specific dependent.
Yep. And then the one other question was, do you have any ideas on why the multi-approach outperforms the weighted approach?
So in general, like we found for PRCS, like this method, we found the absolute accuracy is actually between these two strategies, they are very slight, but usually the meta-analyzed approach is better in terms of the overall comparisons. So the only exceptions for the weighted strategy performs better is that for traits highlighted here, they are less polygenic and there are some population-specific variants. And on the other hand, I need to mention that like for the multi-approach, we need to like use a set of different LD reference panel and choose the optimal one. And on the contrary, like for the weighted combination, we may use ancestry matched LD reference panel. So it's not that affected by the choice of the LD reference panel.
Wonderful. Thank you so much.
Thank you for the questions. So based on those findings, I want to summarize a few general best practices for such polygenic prediction across populations. So first of all, if you want to like derive a PRS based on single ancestry GWAS, like either based on large-scale existing European jewels or a small-scale minority jewels. So it's like dependent or related to the theoretical accuracy I previously mentioned. So if the traits they are with high heritability in both the discovery and the target populations, and the cross-ancestry genetic correlation is high, we recommend to like build a PS based on the large-scale European jewels. But on the other hand, if you have a relatively large sample size of the minority population, and especially like there are some target ancestry enriched variants, then we expect the minority jewels can provide you even better accuracy compared to use a much larger scale like European jewels. But more often, we will recommend you like to utilize these resources from multiple ancestries. So in this study, like we are mentioning the multi-strategy or the weighted strategy. So yeah, so which one to choose is also like dependent on those different factors. And especially like when their heritability are both high in the target population and the discovery population, but when the discovery population is even higher, so the target population can borrow more genetic information from the discovery population. And also this is more suitable for traits with cross-ancestry genetic population higher. So in these scenarios, we will recommend use the meta-analysis strategy, and this is especially true for the method like P+T. But for the weighted linear combination, in this study, we found like two kind of categories for the recommendations. The first one is the trait is with low cross-ancestry genetic correlation, such as height. And also for those traits with target ancestry enriched variants, such as white blood cell counts, neutrophil counts, and for some less polygenic traits like MCH, MCHC, etc. So in this scenario, we recommend use weighted linear combinations. And among all those methods, we found the basic methods generally performed better than the classic P+T method. So if you want to know more details about this topic, you can go to our preprint. And finally, I'd like to thank all my co-authors on this work, and also my colleagues in Martin Lab. Thanks for your attention.
Thank you so much, Ying, for an excellent talk.